Hi guys, and welcome to some video notes. Sorry I can't be there with you guys right now in person, but we're going to go over blood, and you're going to be able to use the information that you learned today to complete the worksheet later on. Also, don't forget to write your questions for our forensic speaker that we'll see together on Friday. And then next week we'll actually do a blood typing lab, and we'll learn a little bit about a very common blood condition called sickle cell. So I think that we have a lot of fun things to do with this topic. So let's go ahead and dive in. So when it comes to blood itself, it has a very long history. I'm going to have these videos linked for you to watch after we
Do you
go
know
through
your
the
blood
slides.
type?
Because I'll get copyrighted on these video notes if I include the videos in my slides. But once again, I'll have those linked to this so that you guys know which videos to watch, and I'll have them in order too. So I would recommend watching through all of these notes and then going through those videos afterwards, just because it's a lot to pause this video, go back to watch the video, pause it, etc. So, a history of blood. We have been aware of the importance of blood for a really long time. We have written history going as far back as 2500 BC, while Egyptians would bloodlet. So that means essentially just make a cut, allow them to bleed in order to cure diseases. And the idea was that you get rid of like the old icky blood, and then your body would make new, healthier blood. And then as we go through, we can see about 2,000 years later, the Greeks identified that we have arteries and veins, and now you know the difference between the two, right? So maybe pause this for a second, chat, what are the differences between those two, right? And then, once you've come up with your answer, come back to the notes. And then once we get into AD, we see a lot more happening when it comes to what is blood, what does it do, what's the role of platelets, how are blood types taken into account, why do those matter, how do we give people blood who might lose a lot of blood, what are some blood transmitted diseases, right? Like AIDS is one of the most well-known ones, but there's also hepatitis and other transfusion transmitted diseases that is carried in our blood. And then we have ELISA testing and then blood screening tests as well. So like I said, you'll have a chance to watch this video after we do all of these video slides, but essentially there's a lot to be said about blood. So when we look at blood itself, what is it made out of? It's made out of primarily plasma, and we'll learn about what each of these four components are in a moment here, but primarily it's made of plasma, and then the second highest abundance are red blood cells, and then we have some platelets, and finally some white blood cells. So plasma is what's going to carry the blood cells and proteins throughout the body. Think of it as like the stuff that then everything else moves in. It makes up about 55% of all of our body's total blood volume, and it's made of up to 95% water, so it's pretty much water. And then the other 5% is going to be proteins, glucose, electrolytes, hormones, oxygen, there's the bell, and carbon dioxide. Now, platelets are also known as thrombocytes, and these aid in blood clotting, and they're involved in repairing damaged blood vessels. So these eventually become scabs. So when platelets sense oxygen entering the bloodstream because of a wound, like a cut or something, they'll gather at the wound to attempt to block the blood flow. They want to make sure that not too much blood is leaving, as well as no potential bacteria, viruses, anything that would make us sick, any pathogens are coming inside. So calcium, vitamin K, and a protein called fibrinogen are what going to actually make the clot itself with the platelets, and then once again, that blood clot on the surface of our skin is going to exist like a scab. Well, it is the scab. Red blood cells are also known as erythrocytes, and the hemoglobin protein that's in our red blood cells contains iron. And we know that our red blood cells bring oxygen throughout our body. It's because that iron binds to the oxygen. These cells carry oxygen from the lungs throughout the rest of the body. They also bring carbon dioxide back to our lungs, and hemoglobin is, the reason that our blood has that red color. And finally, we have white blood cells, also known as leukocytes, and white blood cells fight diseases and foreign invaders. They're part of our immune system, which is very important, and they work to identify cells or molecules that are foreign pathogens, a word I used before, such as viruses, bacteria, and parasites. Once identified, the white blood cells will then engulf or eat and then digest the invader. They also secrete antibody proteins, and they are the only blood cell with a nucleus. So they're the only type of blood cell that can be used to get DNA if you were doing a forensics case or needed to get somebody's DNA from their blood for any reason. So when we look at the different components of our blood here, we can see those erythrocytes. Our red blood cells are going to be the first most common thing outside of plasma, right? Because plasma is like 55%. And then of the 45% that isn't plasma, a lot of it's going to be erythrocytes or our red blood cells. Then we'll have our white blood cells, which are to help with our immune system, and then platelets that'll help with creating a scab or blood clotting. Remember, our erythrocytes, our red blood cells, are going to transport oxygen throughout our body. So what does our blood say? Well, our blood is able to, like I said before, give us DNA from our white blood cells, and it also can tell us our blood type. Blood typing is less expensive than getting the DNA from somebody's red blood cells, but it also is less exact because different individuals might have the same blood type. So this science behind blood typing is really important, especially when we get into blood donations. So let's explore that a little bit. So when it comes to our blood type, this is because of a variety of different proteins that exist on our red blood cells. The main ones that we talk about are AB and our Rh factor. So we're going to go through those today. So having an A or a B or both or neither is going to determine if you have type A blood, type B blood. If you have neither of them, you have type O blood. If you have both, you have type AB. So the A and B proteins are found on the surface of some red blood cells. You might only contain type A protein, in which case you'll have type A blood. If you only have the type B protein, then you'll have type B blood. If you have both A and B proteins on your red blood cells, then you'll have type AB blood. And if you don't have either the A or the B proteins on your red blood cells, then you have type O blood. So essentially, the type of protein that is found on your red blood cell is going to be the blood type that you have. And like I mentioned, we look at A and B, and we look at what's known as the Rh factor. And this is what's going to determine if you're positive or negative. So 85% of the human population has a protein called the Rh factor on their red blood cells. So if you have that Rh factor, then you are positive. If you do not have it, then you are negative. And so we'll say both your AB and your Rh factor together. So for example, I have type A positive blood. That means that my blood has both the A protein and it has the Rh factor. If I had type A negative blood, then I would have the A protein, but I would not have the Rh factor. So if I was type O positive, that means I did not have the A or the B proteins, but I did have the Rh factor. If I was AB negative, that means I have both the A and the B proteins, but I do not have the Rh factor. Does that make sense? We'll have a chance to work with that a little bit more. There's also different things that we can continue to use to look more and more specifically at our blood type, different proteins. So for example, M and N. You might have 2M, you might have N, you might have both. There's, like I said, just different proteins that we can look at to get a more exact blood type, but by and large, we look at A and B and Rh. And this shows you how common they are in the population. So I have type A and I have positive, so I have pretty common blood type. So here's some other enzymes and proteins that can be used to continue to identify blood. We normally don't go this in depth unless you're really sensitive to blood transfusions. Then we might want to focus, on how we can make it as exact as possible. And so as I said before, The name of your blood type depends on your combination of proteins, and they're normally written as the letter and then your RH. So I could be type A positive, right? Or I could be type A negative. And so if this had the RHs on it as well, that would be B positive. How type O doesn't have A or B. And then if type O also didn't have the RH factor, then it would be O negative. But if it did have these, then it would be O positive. Once again, the proteins that you have are the ones that determine what your blood type is. Simple as that. And so when it comes to looking at those proteins, we need to know this information primarily so that when you get a blood donation, it doesn't kill you. And that's because our immune system wants to destroy anything that isn't us. Well, if you get a donation of blood and that blood doesn't match, then your body is going to think you're being infected by something. So we're going to learn now about how that process works, and then finally how to make sure that we're not doing that when we give somebody a blood donation. So before we learned about blood types, transfusions just as often killed patients as it did save the lives as others. So understanding more about antigens and antibodies explains why this happens. So antigens are uniquely shaped bonding sites on the surface of our cells, both foreign and human. So all of our red blood cells, as well as a lot of other cells, have what are known as antigens. These are unique shaped things, proteins, bonding sites, etc., that are on the surface of our cells. Antibodies are Y-shaped proteins that bind to the antigens on foreign pathogens as a way to tag it for destruction. So just think of these as little guards that move through our body, they move through our bloodstream, and these are constantly on the hunt for their match, their matching puzzle piece, because if they make that match, then our body's able to say, whoa, we got a match, that means it's time to get our immune system ready to destroy what's trying to infect us. So the problem is, when that's blood, what our body does is it will take those antigens on the surface of a foreign blood type that doesn't match our own, and our antibodies will bind to all of those antigens to create a clump of blood. And, that causes a blood clot. And what is a big risk for a heart attack? Having blood clots. So white blood cells recognize a foreign substance and try to destroy it in a process called agglutination. Agglutination is the term that we use scientifically to describe when our immune system is reacting to the wrong blood type. Foreign invaders might be pathogens such as viruses, bacteria, or the red blood cell proteins from someone with a different blood type. And so each antibody has 2 binding sites, which means that they can kind of hold on to 2 at the same time. Meanwhile, each red blood cell has about 300 proteins and more than 100 million antigen sites. So essentially, just think of a bunch of little things holding on as tight as they can to as many red blood cells as they can hold together to create a big clump of all of the things that might make us sick. If it's blood, though, that is donated to us to help us not get sick, well, you can imagine why that would then be a problem, because it causes a blood clot. When foreign blood types are introduced to the body, this binding, our immune system reacting to something that doesn't match the blood in our body, causes agglutination or clumping of the red blood cells, which can then create potentially deadly blood clots, which is why a lot of people were dying from getting blood donations, when we didn't really know why it was important that it had to be a perfect match, right? So to identify the blood type of an individual, the patient's blood is separated into 3 separate samples, and then it's mixed with proteins A, B, and RH. And we can use agglutination by taking these blood samples, and then we're going to expose it to different antibodies. And the antibody that causes the blood to agglutinate, or rather, the antibody that binds to the antigens on the blood sample, is going to tell us that blood type. So after this video is done, you can watch this video here, and it'll make a lot more sense as to how we go about that process. But essentially, when you take the blood sample and then you take the antibody, if the antibody reacts, that means that that's the blood type. If it doesn't react, that means that those proteins are not there, which means it's not the blood type. And then finally, if we really wanted to get fancy, we could use information about how much of the population has certain blood types to figure out how rare or common a certain blood type is. So we might use this if I really want to get fancy. Maybe I'll have us calculate how rare something is. But essentially, this is really helpful in forensics, because if you find some blood at a crime scene and then you identify the blood type, the more pieces you look at and the rarer the blood type is, the easier it is to pin the identity of the person that left that blood as evidence. So it definitely has some cool applications. But anyways, those are the slides. Like I mentioned, I'm going to have those videos linked as well so that you guys can watch through those after you're done with this video. And then the information from this video and the slides will be used to complete your worksheet, the blood blends worksheet that's on Schoology as well. And then don't forget to submit your questions to the discussion board for our forensic speaker that we'll be able to watch on Friday. Otherwise, I'll see you guys then. Let me know if you have any questions. Have a great rest of your day. Bye.